Hello everyone. So we are going to start the part 3 from the unit drug stability. So in this part we are going to deal with few more activities which led to the instability of the drug. Apart from that, we are going to discuss related to the kinetics. That is the chemical kinetics, which is associated with the formation of a stable drug product or unstable drug product. So without wasting any time, let's start our presentation. So now you see the type of oxidation. As we have already discussed regarding the oxidation, this oxidation sometimes led to the degradation of the drug, right? So there are different types of oxidation which generally takes place. One, it is your auto-oxidation, other one it is your photo-oxidation. So what is the difference between them? You see auto-oxidation. This auto-oxidation generally takes place in the oil, fat and unsaturated compounds. So the thing is that this auto-oxidation reaction, it generally takes place where oxygen is present in the air. Means if the drug is exposed to an air or an environment or an atmosphere where oxygen is available, the auto-oxidation process tends to take place. This auto-oxidation process is a very slow process, but generally it influences the stability of the drug, right? The next one is your photo-oxidation. So this photo-oxidation, it generally takes place with the drug like adrenaline, riboflavin and ascorbic acid. So in photo-oxidation, you see this oxidation process, it will take place, right? But the thing is that without the presence of atmospheric oxygen. I repeat, without the presence of atmospheric oxygen. That is the definition itself. It's a oxidation is the removal of electron, means it is oxidized, right? Is involved without the presence of atmospheric oxygen, that is O2. Clear? The next is the prevention. So how you are going to prevent the oxidation process? So basically you see, first of all you can use a chelating agent, or you can say, somebody used to say the chelating agent. So you can use a chelating agent, EDTA, ethylene diamine tetraacetic acid, right? EDTA, it can be used to prevent the oxidation. Apart from that, you can use some antioxidant agent along with the drug to prevent the oxidation. So what are the antioxidant agent? Citric acid, tartaric acid, ascorbic acid, isoascorbic acid, salt of actually potassium and sodium of metabisulfite. That is the sodium metabisulfite or potassium metabisulfite you can use to prevent the oxidation. Clear? Next we have microbial degradation. So the thing is that sometimes it is helpful, but sometimes it is not. So the contamination due to the microbial actually of a product, sometimes it causes lot of damage to the product, drug product, and sometimes it may not affect that drug product at all. So thus it is dependent on the type of microbe. There are some microbes which can degrade the product up to a huge extent, and that can produce toxic component from the drug. So if the parenterals means those drugs which are intended to be given in the systemic circulation directly, your injection, salines, ophthalmic preparation, right? So if parenterals or ophthalmic formulations are contaminated, if those formulations are contaminated, it generally causes huge harm in the patient body. That thing we have to keep in our mind. So now you see source of microbial contamination. So from where the microbial contamination takes place? Generally from the water and the air. Where these two things are present, they may be present in the site of preparation at the industry, right? From the raw materials that has been used to prepare the formulation, and from the containers as well as from the closures, this microbial contamination can come. So these are the sources of microbial contamination. Now how you are going to prevent it? You see, first of all you have to suitably design the container. The container should be designed in such a way that these infectious agents which may come from the water or air or raw material, that need to be stopped or that need to be hindered, so that the product stored within the container is stable, right? Next, we generally, for such cases, we use a single dose container. So what are single dose container? In a single dose container, we have a single dose only. For example, the blister packaging of tablets. For blister packaging of tablets, at one time we can take out only one tablet from the strip, right? Those are single dose container. Now, apart from that, we have to, or we should stick to the proper storage condition. If the condition is mentioned there as stored in a cool temperature, or stored away from the light, or stored in that particular temperature, then we should maintain that particular storage condition that has been given or instructed by the manufacturer, right? Apart from that, we can use some antimicrobial substances as preservative in that particular formulation. Okay, so now the next concept is the therapeutic degradation and toxicological degradation. So what are these? So you see, the therapeutic effect of any drug must be changed due to the process like hydrolysis, isomerization, epimerization, regarding which we have already cited few examples. We know that, right? So example is your adrenaline, right? So some drug may produce toxic product also, that is known as toxicological degradation. Example is your tetracycline and chloramphenicol. These two drugs generally produce toxicological products upon degradation. So this type of degradation is known as toxicological degradation. Now let's see the concept of kinetic stability. So what is kinetic actually? First of all, the thing is that what is kinetics? The kinetics actually, it deals, or it is the study of rate of reaction and the mechanism. Kinetics is a study of rate of reaction and the mechanism. So actually in the kinetics, what we used to study is, you see, in the kinetics, it involves the study of the rate of change and the way in which the rate is influenced by the concentration of the reactant or product, or by some any other attributes which include chemical species or physical factor like presence of solvent, pressure and temperature. So that is the overall kinetic. So we should have a proper study or proper documentation or proper information of the kinetic stability of a drug before transporting the drug or before formulating the drug, right? So this kinetic actually applies to a wide range of area, which include, number one, stability. In stability, we use or we study the kinetics, right? In the incompatibility study, we study the kinetics. In the dissolution, we study the kinetics. In absorption, we study the kinetics. In distribution of the drug, we study the kinetics. In drug action at the molecular level, we study the kinetics. And finally, at the, even in the process of elimination process, right? So now, what is rate and what is order of a reaction? So rate of a reaction means the speed with which the reaction will take place, and the order, we know what is order, right? So basically, rate means the speed or velocity of a reaction at which the reaction will take place, right? So it determined by the change in the concentration of the reactant or product as a function of time. So it is generally denoted as what? D by dt, means D, that may be X also, that may be Y also, that may be A also. This X, Y, A, whatever it is the concentration. It is the concentration. D small d, it is the change in the concentration with respect to time. That is dx by dt, means change in the concentration of the X drug, small change in the concentration of the X drug with respect to time, right? The next concept is the order of the reaction. Now, what is order of the reaction? It is nothing but the number of concentration that determine the rate. The number of concentration that determine the rate. Suppose we have a reaction A plus B is equal to C. Now, if the rate of the reaction it is controlled by both the concentration of the A and the concentration of the B, we will call it as a second order reaction. If it is affected by only one, we will call it as what? First order reaction. It is free. If it is free from the concentration of the A and B, it is zero order reaction. Then sometimes there is one more order, that is a pseudo first order reaction. In pseudo first order reaction, what we will get, we are going to get a mixed type of reaction. Earlier sometimes it will show a first order reaction, and later on as the reaction is processed it may show zero order type of reaction. So what is zero order reaction? The rate is constant. If A plus B is equal to C, if the rate is continuous, it is not dependent upon the concentration of A or B, we used to say it as a zero order reaction, right? For first order reaction, the reaction rate of change is proportional to the drug concentration, that is either your A or B, right? Second order reaction, the rate depends on the product of the two concentration terms. It will depend on both the concentration of A and concentration of B. And in the pseudo first order reaction, as I already told you, it is a mixed type of reaction. Means sometimes it will show zero order reaction and later on it will show first order reaction, or initially it will show first order reaction, later on it will show zero order reaction. Clear? So the overall order of the reaction, this is the chart. So for zero order reaction, rate is equal to K naught. First order rate is equal to K1. Second order rate is equal to K2. Right? A is the concentration of the reactant. Right? The concentration time equation is this for zero order, for first order, for second order, and this is their half-life. We know that, right? And the plot, graphical plot, it is generally plotted against this concentration versus time for zero order reaction, log of the concentration of the drug A versus time, it is for first order, and inverse of the reactant versus time for the second order reaction. Clear? Now you see self-life. So what is self-life? Self-life is nothing but, suppose we have a drug A, right? Initially the concentration of the drug in the reaction before taking place, before the reaction will take place, initially the concentration will be 100%, right? Now the reaction will proceed till the time 90% of the drug has been used. That is the self-life. So you see, it is defined as a time, the time needed for the concentration of the reactant to reduce to 90% of the initial. 90% of the initial means earlier the concentration of the drug was 100%. After time t, suppose 30 minutes, the concentration has come down to 10%. Now it has decreased by 90%. Means 30 minutes it is the half-life, or you can say it is the self-life actually. So it is generally represented by self-life, it is represented by T90, because 90% of the drug has been degraded. So unit for the self-life, it is time per concentration. So the reaction is T90 is equal to A minus 0.9 A divided by K naught, which is equal to actually 0.1 A by K naught. Here A, it is the initial concentration of the drug, it is 100%. K naught, it is the specific rate constant for the zero order reaction. So these data are generally put in the graph to check the concentration or the stability or the self-life of the drug at different temperature, 40 degree, 50 degree, 60 degree, 70 degree. For every temperature, the rate of the reaction will be different. The rate of the reaction will be different, right? The next concept is the solution stability. So what is solution stability? It is nothing but the stability study of the solution. Stability study of the solution. So the main purpose of the solution stability is the identification of the condition in which we are going to get a stable solution. We generally perform this solution stability for the liquid dosage form, right? Now, what are the conditions we are going to identify? Identification of the condition necessary to form a stable solution, right? This condition includes actually effect of the pH, ionic strength, co-solvent effect, light, temperature, oxygen, whatever it is, right? So interested experiment, for this type of experiment we used to take extreme condition of the pH and temperature, like 0.01 normal of the HCl. We are going to take water along with sodium hydroxide 0.01 normal, right, at a temperature of 90 degree. And for maintaining this pH, generally aqueous buffers are used to provide a wide range, right, wide range where constant level of the drug, co-solvent and ionic strength is maintained. It is maintained and it is generally compatible. This buffer, they are generally compatible with the physiological media. where it will act. Means if the drug is supposed to be act in the lung, then we know that what is the fluid which is present in the lung, what is the pH of the fluid which is present in the lung. According to that only we are going to take the buffer, we are going to make the buffer, right? The next one, it is your solid state stability. Solid state stability study, it deals with the solid state drug. Solid state drug means the drug in the out of formulation in the solid state. For those type of formulation, tablet, capsule, we used to perform the solid state stability. So you see the purpose of this solid state stability is the identification of the stable storage condition for the drug in the solid state. Tablet we have made, now we need to study the stability. So here we are going to consider the physical attributes which will affect the stability of the solid drug, right? And identification of the compatible excipient for a formulation. We should know, suppose for a tablet we have used the starch as a additives. Now this starch, it is showing some effect in the solid state. In such cases we have to remove this starch with the lactose, right? This sort of study they are generally performed in the industry before formulating a solid dosage form, right? So this is affected by the change in the purity and crystallinity of the drug. If the purity and the crystallinity of the drug is changed, it will affect the solid state stability. Next, initial bulk lots and newer lots. It needs to be studied before formulating. Means initial bulk lot. Bulk lot means the bulk lot of the excipient or the drug, and a newer lot before making any formulation. Next, the solid state, actually it is slower and difficult to interpret than the solution state, because the formulation it remains in the solid state. You know that, right? And we use some techniques like TLC, thin layer chromatography, UV visible spectroscopy, and fluorescence study. We perform all this sort of study to check the solid state stability of the drug. Apart from that, polymorphic changes may occur in the drug, and this can be studied using the DSC, that is the differential scanning calorimetry. Even you can use IR, that is the infrared FTIR, actually Fourier transform infrared spectrum, or its appearance, because polymorphism, when it takes place, we can see the changes in the appearance, right? Apart from that, change like oxidation. If oxidation has takes place in the solid state drug, then change like oxidation, it can be easily detected, like surface discoloration may take place, fading of the color of the solid may take place. Clear? So the last one, it is the pH stability study. So in the pH stability study, you see this pH stability profile, it is very much essential for understanding. We need to understand how the compound will behave in different environments. We are going to make a compound, right? Suppose we are going to make the tablet. This tablet, once it will enter our mouth, it will go via the esophagus into the stomach. In the stomach, it is going to reach acidic means acidic condition. Afterwards, from the stomach, if it reaches the intestine, there it will reach first of all it will reach a neutral condition, then it will reach a alkaline condition. So in different pH, actually in different pH, acid, neutral, alkaline, in different pH, the effect and the stability of the drug, it is actually studied using the pH stability study. So how the compound will behave in different environments, and informs the formulation development team and the process development team related to the study of the drug in different environment, and it will actually determines the drug stability, as well as it will help to determine the route of administration. Suppose, for example, I am giving you protein. Now we know that the different form of protein can be taken as a drug, but you say insulin, right? We cannot take insulin via our mouth, because insulin is a protein. If you take insulin via our mouth, once it will reach the stomach, in there, what is the condition? That is it is highly acidic. In the highly acidic condition of the stomach, this insulin, the structure of the insulin may get distorted, and it can lose its effect. In such cases, we have to give the insulin directly into the systemic circulation. Actually, this pH stability study, it deals with the study of the drug in different pH. Means the drug as it is going to encounter different environment, so how the drug will act in the different environment. The data it is will be gathered from this pH stability study. So to develop a pH stability profile, it is very much important to develop the stability, that is indicating the assay for which the drug is intact at the various pH value need to be studied. So before administering the drug in the patient, first of all we have to determine the route. Once we will determine the route, then we should know what are the different conditions the drug is going to encounter, suppose acid, alkaline, etc. So in different pH, acid have a different pH, alkalinity have a different pH, right? So in different pH we have to see the stability of the drug, right? So thank you everyone for your patience sharing. If you like the video, just press the like icon. You can share this video and you can subscribe to my channel. Thank you once again.